Thanks everyone for joining. I'm pleased to be able to share some of the recent advances our lab has made towards more in-depth ADC characterization and what we can provide as part of our mass spec lab services. Okay, so just a quick overview of what I'll be discussing in this webinar. I'll first briefly touch upon antibody discovery and how we perform antibody sequencing. Then I'll go into some considerations that need to be made for antibody-drug conjugate development. And then I'll provide an example of an ADC that we've analyzed in our lab, which is trastuzumab emtansine. And I'll go into how we calculated the drug to antibody ratio, localized conjugation sites, and perform proteoform analysis by multi-level mass spec. And lastly, I'll just highlight a couple publications that came out of a collaboration our lab had with an academic collaborator from McMaster University. Okay, so prior to developing an ADC, you obviously have to discover a novel antibody that binds your target of interest, whether it be an antigen on the cell surface of a cancer cell or some other foreign invader. So the antibody discovery process typically starts with immunization of an animal model, followed by isolating the B cell population and creating hybridoma cell lines by fusing to melanoma cells. And this first workflow on the left, this process involves isolating individual hybridoma clones, screening them for the desired binding affinity, and then performing the antibody sequencing. So the discovery part and retrieving the sequence would then allow for that antibody to be reverse cloned and expressed and batch produced. There are other methods that involve next generation sequencing techniques where you can sequence the whole population of B cells. However, this can often lead to some discrepancies in not knowing which heavy and light chain pair together. And also you can isolate antibodies directly from serum by pulling down with the target antigen and then identifying those antibodies by LC-MS. So our lab typically would come into play at this last step in retrieving these unknown antibody sequences by LC-MS, as well as sequencing monoclonal antibodies derived from these hybridoma cell lines. There are other techniques I haven't mentioned here, but I won't go into detail on those, such as phage display and yeast display methods. So a quick overview of our sequencing service. So once we receive an antibody sample, we perform multi-enzyme digestion to generate different lengths of peptides. And then once we've run these digest on our high-resolution LC-MS instrument, we input the raw data into our PeaksAB software, where the raw spectral will be de novo sequenced to derive the peptide sequences. And then these peptides will be assembled into complete heavy and light chain protein sequences. Once we've assembled the amino acid sequence, we validate that we have the correct sequence by matching to the intact mass measurements within certain error tolerance. So this service, we provide 100% sequence accuracy from N to C terminus for heavy and light chains and intact mass validation, as I mentioned. And from the time we receive your sample, we have the reports ready and sent back to you within 2 weeks. And then if you're interested, we have an optional in-depth glycan profiling as an add-on to this service. So a little bit more about sequence assembly. So after doing de novo sequencing within PeaksAB, we filter to include only high quality de novo sequence tags. And those passing our scoring criteria will be used in the assembly algorithm. So basically the software goes and looks for amino acid sequences that overlap at either N or C terminus of each peptide and continues building what we call this tiling path until you've reached the ends of the protein. as part of the service, we can discriminate between leucine and isoleucine residues, and we primarily rely on ETHCD data. So during this process, we perform ETD fragmentation to dissociate the side chain of the amino acid. And then we look for the mass difference between Z and W ions after side chain dissociation. So the mass difference between those ions for leucine would be 43, and for isoleucine would be 29 Daltons. And we'll annotate these on each of the MS2 spectrum. This glycan profiling result is included in our standard report. So we can retrieve all the glycans and N-O-link glycans actually that are attached to heavy or light chains, and we'll report all those glycan moieties. The optional add-on to this service is if you want more in-depth glycan profiling, we can do things like site profiling, where we report the relative abundance of different glycans, as well as sample profiling, looking at relative abundances of the same glycan across multiple samples. And in that add-on service, we would include the quality spectrum of the glycopeptides that support each of those identifications. And then lastly, our software will automatically match the amino acid sequence. So it'll calculate the theoretical mass from the light and heavy chain amino acid sequences, and then compare it to the measured intact mass while considering certain common modifications such as glycosylation, C-terminal lysine truncation, and N-terminal pyroglue from glutamine. Okay, so moving on to ADCs now. So an ADC obviously consists of the antibody protein as well as a linker, which is conjugated to a cytotoxic drug, which is often referred to as the payload. So the goal of developing an ADC is to have the molecule target a specific antigen that's located on the cell surface, for example, of a cancer cell, and deliver that payload after being internalized within the cell. And after antigen binding and this receptor-mediated endocytosis, the ADC product will be degraded and broken down by the lysosome, and then the payload essentially is released to the cell where it elicits its cytotoxic effects. Okay, so some considerations when doing ADC development from a pharmacology perspective, and these are guidelines outlined by the FDA and ICH, where you have to consider, after doing conjugation and analyzing site specificity, you have to consider how that conjugate may affect antibody binding affinity, the stability of the antibody. It may interfere with internalization of that ADC, and ultimately affecting drug potency, effective dosages, and the safety and efficacy of the drug. And there have been a number of reports, I've just highlighted a couple here, demonstrating that location definitely matters. So where that conjugate is attached can definitely affect the stability and pharmacokinetics of that ADC. And also where the drug is attached may affect whether that ADC will be internalized by that targeted cell. So that being said, it's important to perform in-depth characterization of these ADC products during development to ensure you're producing a drug with the desired activity. And so one of the challenges is, depending on the type of conjugate and the type of conjugation chemistry being done, the linker could be attached to different amino acids. And so early on in ADC development, certain chemistry was used that would attach the linkers to basically any lysine throughout the antibody or any exposed lysine residues. And also this can be done by targeting cysteine residues. So given the fact that there are a number of possible conjugation sites, a number of lysine positions throughout the antibody, you generally get a very heterogeneous mixture of different ADC products after conjugation. So you could have anywhere between 0 linkers attached to up to 8 if you're conjugating through lysine residues. And for cysteine, it's a little bit more specific and typically targets the hinge region of the antibody, but again, you still have somewhat of a heterogeneous mixture. And so not only do you not know exactly how many different products might be in that solution, you don't know the location of these conjugates per ADC product. So for example, an ADC with 3 conjugates, within that population, you could have various different conjugation site attachments of that linker. And so I'll get into that a bit later about how we characterize the conjugation sites associated with each of these ADC products using top-down mass spec. I just wanted to mention somewhat more recent strategy for more site-specific attachment of the linker and payload. And that's using this technology called genetic code expansion. So essentially, when expressing the antibody where you want that linker to go in, you would designate a stop codon. And since the stop codon doesn't encode any other amino acid, you can engineer a tRNA to specifically recognize that stop codon, and it should only go in at that site. And that tRNA is ligated to this chemical handle that serves as a linker for conjugating the payload to. And so in doing so, you can have confidence that that linker is only going in at that specific site where you've designated the stop codon to be. This still does have some drawbacks. It's can be a fairly inefficient process in terms of the yield of antibody that you can get from these cell lines. These expression systems tend to be fairly toxic to the cell, and the cells don't grow so well. But nonetheless, it is very effective at attaching that payload to the desired site. So our solution to analyzing these ADCs is to combine multi different levels of mass spec, so intact mass, bottom up and top down analysis to have a more thorough understanding of what proteoforms might be within that conjugate, within that sample. And so in doing so, on the left here in part A, we'll analyze the intact mass of each ADC product within a sample. And then that will allow us to calculate that DAR. Using bottom-up proteomic approach, we can have confidence in localizing the conjugation site within that ADC. However, this won't tell us any information on how many conjugates are within one molecule. And that's where top-down comes into play. So we can keep track from by injecting the intact protein and recording the intact mass prior to doing fragmentation. We can then build back up the top-down fragment ions to match the intact mass of that ADC product, all while identifying the conjugates and determining how many conjugates are part of the same molecule. And so here's a bit more detail on our strategy. So intact mass measurements will be used for the DAR calculation. But again, this doesn't tell you the site localization or where that linker is being attached to. And we'll combine that with bottom up. So bottom up's very accurate and sensitive at localizing that conjugation site to specific amino acids. But because you've digested the proteins within your sample prior to running them on the mass spec, you've lost the information of which of these proteoforms each peptide was derived from. So this is why it's important to integrate top-down or middle-down approaches in order to actually characterize the population of different ADC products within a sample. And so what I'm trying to illustrate here is that it's important to do this because you could have various different ADC products that have, for example, 3 conjugates attached to that antibody. However, based on the location of each of these conjugates or the conjugation sites, you may have some of these products that are ineffective at, say, binding the target antigen or being internalized by cells. So it's important to not only characterize the number of conjugates on the antibody, but also determining, say, for example, an ADC has 3 conjugates, where each of those conjugation sites are located. And so to demonstrate this workflow, we purchased this ADC from Thermo called trastuzumab emtansine. So it's well characterized. It's a highly heterogeneous ADC product, since it undergoes the payload undergoes lysine conjugation. And so the bottom left here displays the linker, which is referred to as MCC, and the drug, which I'll refer to as DM1. And so first I'll show some intact results and how we more accurately characterize the drug to antibody ratio by analyzing native intact mass of the sample. And then I'll show some bottom-up data and how we accurately localize number of conjugation sites throughout heavy and light chain. And then lastly, I'll go over some preliminary top-down and middle-down data that we've recently generated and how we're characterizing the different proteoforms within that sample. So here's our sample preparation workflow. So we take the trastuzumab ADC. The intact preparation is fairly straightforward. We either do no treatment at all, just a desalting step, or we'll also take it and do reduction and deglycosylation to try and simplify the deconvolution of intact mass. For top down or middle down, we treat it with IDS that cuts at the hinge region, releasing the FC, the constant region, and also perform reduction deglycosylation prior to running it on the mass spec. And the bottom up is a multi-enzyme digestion method after performing reduction alkylation and then running these products on the mass spec. So first, starting off with the intact analysis that we've done. So initially we tried just to deglycosylate the antibody and then run the intact form of that product. So here's the chromatogram shown in the middle, and we had a sliding window acquisition from this time frame in order to deconvolute the peaks shown in the higher mass range here. And from that deconvolution, here's just zooming in on that mass range, we can see that we've identified a number of different conjugated forms. So going from the unlabeled antibody all the way up to a total of 6 DM1 molecules conjugated to one antibody. But as you can see, the DAR is quite low, and it was a lot lower than we expected. So kind of hypothesized that the treatment or deglycosylating the antibody and doing this overnight deglycosylation step and incubation may have led to some loss of conjugated products. So we decided to perform a more mild sample prep procedure and just basically doing the desalting step and no further incubation or steps after that. So again, here's the chromatogram and the peaks that were deconvoluted in that range. And as you can see, we have a number of glycoforms in this case. Obviously, we haven't removed the glycans, but in fact, the products that have 2 or 3 conjugates are in much higher abundance compared to the deglycosylated sample that we prepared previously. And actually, we calculate this average DAR to be 2.4, so more than twice that of the deglycosylated sample. So in conclusion, it's best to use a mild treatment in analyzing the native intact mass when performing the DAR calculations. So next I'll show the bottom-up analysis we did. So we digested the ADC with multiple different enzymes and then map those to the trastuzumab heavy and light chain sequences. So all the modified peptides, you can see a colored square with the lowercase d representing the DM1 conjugate associated with that peptide. And here I'm showing the triptych peptides mapping to heavy chain sequences, and we identified these 5 different lysine sites conjugated to DM1 or conjugated with DM1. And this is why it's important to use multiple different enzymes when doing bottom up. It's because with pepsin, we identified 3 additional sites of conjugation at K277, 395, and 417. So we seem to get better coverage and more modified peptides being identified in the FC region using pepsin. Oops, sorry. And then lastly, we also digested with elastase and chymotrypsin and identified conjugation at lysine 213 and 291. And for the light chain, this is just a combined view of all those digests, different digests. And in the light chain, we identified lysine 42 as a conjugation site, in addition to lysine 107 and 126. And further towards the C terminus, we saw more extensive conjugation at lysine 145, 183, 188, and 190. And I will point out there's other labeled or modified peptides shown here. These represent just the linker where there was no DM1 drug conjugated to that linker. And here's a representative spectra, MS2 spectra, showing both the unconjugated and conjugated forms of a peptide from the sample. And I just wanted to highlight how we accurately can localize that conjugation site from bottom-up data. So we apply different scoring criteria and ensure that we have good fragmentation of the peptide backbone in order to differentiate between, so for example, this peptide has 3 different lysines, but we can be confident that this third lysine is the conjugation site since we have evidence from peptide backbone fragmentation, fragment ions to support that. And also as further proof that this is the conjugated form, at the MS2 level, we also get fragmentation of that linker and DM1 molecule. And so we can look for these kind of signature ions that are associated with this modification. And here we can see a fragment ion peak at 547 Daltons, which corresponds to the drug portion of this conjugate. Okay, so in summary, after bottom-up analysis, we identified a number of lysine conjugation sites across heavy and light chain in a total of 15 different DM1 conjugation sites across the entire antibody. So next I'll show you some of the preliminary results we have from top or middle down analysis. And I don't know if I mentioned this earlier, I refer middle down as those products generated from IDS digestion, and basically when the heavy chain is being cut at the hinge region, we refer to those domains as middle down products. And so we input the heavy and light chain sequences into our software, and here you can see the number of possible lysine positions highlighted in pink across heavy and light chain. So we go in and search for modification of the DM1 linker at each of these sites, and also consider possibility of disulfide bonds when calculating the mass of each proteoform. So starting with the heavy chain, here's the FD domain, which essentially the Fab or antigen binding fragment of the antibody generated after IDS digestion. And so we do get fairly good sequence coverage from this top-down data of that FD domain in the heavy chain. And by sequence coverage, I mean the percent of fragment ions that are generated from this intact protein. And it's important to have reasonable sequence coverage or good fragmentation in order to distinguish between all the different lysine sites across each antibody domain. So with the unlabeled FD domain, we did see very good sequence coverage. And also for the FC region of the heavy chain, we saw exceptionally good sequence coverage and good top-down fragmentation of the heavy chain, which would allow us to distinguish most of the lysine residues and help to localize each conjugation site. However, with the conjugated product, what we're seeing was quite lower top-down fragmentation and lower sequence coverage. So this did leave us with some ambiguity since we don't have fragmentation between a number of residues. We don't know for sure, just strictly from top-down data, whether DM1 is conjugated at this lysine versus this lysine. But nonetheless, we were able to confirm a proteoform of the FD domain with one DM1 molecule conjugated to it, as well as the FC domain of the heavy chain with one conjugate associated. And then also we did see a fragment of the FD domain, which was predicted to have 3 DM1 molecules conjugated at various lysine residues. And with this data, the goal is also to look at relative abundance of each form. And so we can report the abundance values since we do feature detection of these top-down from top-down data. We can associate abundance values with each proteoform. And for the light chain, again, we have fairly good sequence percent coverage and fragmentation of the unconjugated form. However, this percent sequence coverage is a bit lower when we've identified a conjugated form. But here is an example of how we're able to localize DM1 conjugation to this particular residue. And ultimately, I've highlighted it, outlined in the dotted squares are the different domains that we ran for top-down. And from this is kind of a summary of the top-down results, and these are the potential proteoforms that we've identified from that data. And so you can kind of combine the bottom-up data with this top-down data now to try and more accurately localize those sites and regions where you have poor top-down fragmentation. But our next steps within the lab now are to move ahead with trying different ways to improve top-down fragmentation and sequence coverage. Okay, and lastly, I'll just mention a couple papers from the lab that we collaborate with at Anthony Rouleau's lab at McMaster University. So their lab develops these antibody recruiting molecules. And basically the way these work is shown in the image on the left here, where if you'd like an antibody to target an antigen displayed on the cell surface of, say, a cancer cell, you can more effectively target immune effector cells or immune response to these cells by conjugating that antigen to the antibody, particularly in the antigen binding fragment, and have it associate more stably with the target cell in order to improve recruitment of the immune effector cells. And so they've published a number of papers on this technology that they've developed, and they've used our lab services in order to validate that the antibody has been conjugated and to localize the conjugation site. And so we were able to do this for them. So you can see this one molecule that they conjugate by SUF-X reaction will target tyrosine residues. And we had in-depth coverage of these modified peptides corresponding to the conjugated peptide form, all mapping to lysine 34 in the light chain of the antibody. And then by using this different type of C-arm, they were able to conjugate different molecule to lysine 55 of the heavy chain. And so again, we have high confidence in the localization site supported by this bottom-up data. And we can further support that by top-down data, which is shown at the bottom here. So in summary, I showed that native intact mass analysis and using more milder sample prep treatment is best for calculating drug to antibody ratios. Top down and middle down can reveal different proteoforms, and those proteoforms can be associated with abundance values. And bottom up approach is valuable for the accurate localization of these conjugation sites that were identified or from the different proteoforms. This workflow can apply to essentially any ADC sample, in addition to the other examples we've shown involving antibody recruitment molecules. And lastly, our ADC analysis can benefit early phase development by informing on ADC potency and effective dosage, which will become very important for later clinical development. So with that, I'd just like to thank everyone for listening, and I'm happy to take any questions.